Hello, howdy. My name is Eric Villasenor, and it's getting hot in here with PIF4 and temperature regulating immunity and disease, chlorosis and Arabidopsis. So a little bit about me. I got my first degree in physics at the University of Miami in 2022, and I'm currently at Virginia Tech doing a dual degree in environmental horticulture and plant science. Virginia Tech is also where I'm doing my home research, where we're basically truncating our response factor and trying to put synthetic interaction domains on that to elucidate the auxin signaling pathway. Super exciting stuff, but that's not what we're talking about today. But what motivates me? I think the biggest challenge our society has to face is how big our society is in terms of population. And it's exploding. It's getting more and more every day. What does this mean? It means a lot. It has a lot of downstream effects. People are the #1 driver of climate change, getting hotter, getting colder, extreme weather patterns. We're also introducing new vectors for disease, and our practices, our pollution is creating disease pressure on our crops. And speaking of agriculture, 70% of our fresh water use goes towards agriculture. So that's decreasing our amount of fresh water. And with the greater population housing that urban expansion, that's decreasing the amount of arable land we have to feed that growing population. It's crazy. So all of this at the end of the day just means big financial and agricultural losses. Nobody wants to lose money, right? But specifically, my research is about PIF4, and PIF4 actually lies in this nexus here between climate change and disease pressure. So what is PIF4? Well, PIF4 is a transcription factor. Yay, more transcription factors. Specifically, it is regulated primarily by phytochrome B, which is a photosensor and thermosensor. So in a nutshell, PIF4 responds to light, responds to temperature to regulate other genes of interest. How does that look? Well, it looks something like this. We have 5B here, and in its active form, it is clamped on to PIF4. However, at high temperatures, it reverts to an inactive form, freeing up PIF4 to directly bind to promoter sites in DNA, thus blocking transcriptional machinery. So, what is PIF4 in regard to my experiment? Well, we know that PIF4 is a positive regulator of growth at higher temperatures, but it is also putatively a negative regulator of immunity. I say putatively because there's some debate on this point. The Gongapa lab says that there is definitely a correlation of negative regulation of immunity by PIF4. However, the Huat lab is like, no way, Jose, this has nothing to do with immunity. And the two differences between those two studies are methods of inoculation. So the Gongapa lab did a dip spray inoculation and saw significant results. The Huat lab did a direct injection, saw no results. So maybe this has to do with stomata, because if you spray the inoculate onto the leaf, it has to enter through the stomatal surface to get to the mesophyll. So our lab was like, well, let's see if that's true or not. This was actually done before I showed up to see, like, do we want to do this further? We have a spray test here and an infiltration test here on the left. And as you can see, there is significant difference in infection levels when the bacterium has to enter through the stomata. So we're like, okay, that looks good, let's do it. So what am I talking about? I'm basically saying that PIF4 is potentially a negative regulator of immunity when instigated by bacterial crossing the stomatal threshold into the mesophyll. What is a stomata? You've all been wondering this, right? I said it like 5 times. Well, it's this little pore right here in the leaf surface, usually on the underside of the leaf. And it's a pore for facilitating gas exchange. So like a plant will breathe in carbon dioxide, it will breathe out oxygen and water. And that happens through the stomata. So we're saying that bacteria going through this little hole in the leaf is what's prompting this response by PIF4. How do we measure that? With many, many assays. So we're doing growth assays, we're doing disease symptom assays, we're doing immunity assays, we're looking at stomatal aperture. It's a lot, but a very big component of my research was actually just getting the genotype lines to study specifically the effect of PIF4. So we have 4 main genotypes. The first one is wild type, Columbia 0, we all know that. The next one is our null mutant, PIF4-1, which does not have PIF4 expressed anywhere in the plant. Next, we have the PGC line, which stands for promoter and guard cell. And that is basically in the null mutant background. So we have a null plant, and then we reintroduce the PIF4 at promoters for guard cells, so it's only in the guard cells. The little stomata right there. And finally, we have a PPIF plant, which stands for promoter for PIF. It's native promoter. So we have a null mutant, completely get rid of it, and then we reintroduce it to its native promoter. And essentially what that looks like in practice, it's kind of like an overexpression line. So here's the first growth assay, hypocaudal lengths. And as you can see here, it kind of confirms our results of what we thought it would look like. The wild type is significantly different from everything else. The overexpression line is significantly different. And the promoter for guard cell is the same as the null mutant, which is expected because if you have PIF4 in the guard cell, that shouldn't affect hypocaudal, right? A hypocaudal, by the way, is the part between the seed root and the cotyledons. So that's what we're measuring, that little length right there. So overall, we can see that in comparing the wild type to the null mutant, we see less overall growth phenotype, which is, consistent with PIF4 being a negative regulator of growth. Because PIF4 is gone, it grows less. I mean, positive regulator, yes, sorry. And then we can also see that PGC mutants don't contribute to the overall growth phenotype, which is expected. Next assay, petiole length assay. And that basically confirms the hypocaudal length assay. It has the same exact trend, right? Yes, good, okay. Next one. We have disease symptom analysis. How do you quantify disease symptom analysis? Well, a big symptom of bacterial infection is chlorosis. So you inoculate the plant, you let it sit there for a couple days, and then the leaves are like, oh no, we're infected. We got to die off and get rid of this infection. And that happens through chlorosis and eventually necrosis. So we're trying to put a number on that, which is why we did a 80% acetone chlorophyll extraction to try and quantify the levels of disease symptom in response to our inoculation. Overall, I would say all we can really glean from this is that at higher temperatures, there is more chlorosis. Surprise. Stomatal aperture. This is where it gets really interesting. We're like, okay, if the PIF4 is happening in the guard cell, then it must be maybe affecting guard cell opening and closing, AKA stomatal aperture. So perhaps this method of immunity is just the stomata being more closed, not letting in as much bacteria, therefore it has more immunity. We tried to test that out and we got some significant differences between wild type here and our null mutant PGC. However, the PGC line was behaving in a strange way. The 22° was consistent with the null mutant, but the higher temperature was consistent with our wild type. Why might this be? Well, I think that this has to do with its master regulator, 5B. As I mentioned earlier, 5B is inactive at higher temperatures, allowing for more PIF4 expression. And when you have more PIF4 expression, it's going to restore the wild type phenotype, rescue the phenotype. So basically, that looks good. What just happened? Well, at the end of the day, we can say that PIF4 is a positive regulator of growth and partially, potentially a negative regulator of immunity, but it's hard to say. We can say that it happens specifically in guard cells, the response by PIF4, but not necessarily in the overall growth phenotype. And we can also say that there's a direct correlation between PIF4 activity in the guard cells, according to temperature. Future directions, maybe do the chlorophyll assay again, get some significant figures. We also want to look at more disease response through a CFU analysis. What does CFU stand for? It stands for colony forming unit. You basically extract the bacteria from the mesophyll and then dilute it to a point where you can count individual colonies on a plate like this to estimate the level of infection. And I would like to thank my lab, Dr. Hua, and my mentor, Veronica Perez. And I really like to thank my cohort also because you guys are awesome. Thank you. Any questions? Yes, Dahlia.
That was wonderful.
Thank you.
Another thing I'm curious about, I know your experiment focused mostly on temperature
Mhm.
as a function of like climate change, but we also know that weather patterns are also changing and that humidity may also be changing. Do you believe that the PIF4 gene behavior that you see with temperature may also change with changing humidity, depending on like different regions for bacterial
That's a very good
infections?
question, Dahlia. Well, when is it more humid? I would say maybe it's more humid when it's raining. And when it's raining, it's cloudy, so there's less sunlight. And as you know, phytochrome B is a photosensitive and thermosensitive regulator of PIF4. So less light, less things happening with PIF4. So maybe it is kind of dependent indirectly to humidity as a function of light.
Thank you.
Another question?
Hi, thank you for your talk.
Thank you.
I'm wondering, how did you make the mutants? What kind of technology did you use? Do you use CRISPR-Cas, by the way?
Oh, it's just regular breeding. I was not able to be here when they made the null mutant, but I can tell you that you go through like 3 generations of breeding to make sure that it's homozygous. They probably knocked out the first gene and then reintroduced it as specific promoters. I don't know. I was more focused on the assays for my project specifically.
Thank you.
Thank you. I think it's exciting personally, PIF4 being at the nexus of temperature and disease, like that could be huge for our crops. If we are able to not have plants express that disease phenotype, that's just more to sell on the fresh market. That saves farmers money. That saves everyone a heartache. I think it's great. Yes,
Eric,
Jade.
what was your favorite part about your research? Like, what was the thing that you were doing in the lab where you were like, oh my God, this is so fun? Or just
I really liked being in a new lab culture, meeting new people, learning new techniques. I think it was great for me to experience the new, so that way I could really reflect on the future, my future personally, if I want to do grad school, if I want to do this research or in a different field. And I did learn a lot about myself, and I learned a lot about lab culture different from my own. And that was my favorite part about the experience.
congrats, Eric. I remember the first day you were like just talking about how you wanted to be successful and like that's what you were striving for. And I think you did a good job.
Thank you.
So, wait, you're not done yet.
Oh, okay. I got preemptive. Sorry.
You talked about Arabidopsis. Are there PIF4-like genes in other plants? Would this be relevant to corn or beans?
Yeah. In my abstract, I was looking like, who cares about Arabidopsis? We want to see other things. And it is a big part of rice. And I think it's also corn, yes. And who knows what else? There are endless possibilities. We just need to investigate it, right? But I think it's very relevant to our major crops today, staple crops used around the world to feed billions of people. Yes.
Okay, thank you.
Thank you.